everybody. All right, I'm going to start by introducing our Crick lecturer today, Sir Shankar Balasubramanian, who is a chemist by training. He was born in India, and then he moved to the UK when he was very young, just a year old with his parents. And as I understand it, he grew up in Cheshire. And then he did his undergraduate degree here in Cambridge, where he then also continued to do his PhD to study enzyme mechanisms with Chris Abel. He then did a short stint, a couple of years in the US as a postdoc at the University at Penn State with Stephen Benkovich. And then he came back to Cambridge to start his own lab, where he then did, I think, quite an interesting sort of turnabout and became very interested in nucleic acids. And this is then where he developed a groundbreaking new technology called Solexa sequencing, which would eventually, of course, become what is used by the vast majority of us today to do next-gen sequencing. And so that was then acquired by Illumina, and that's what many of us here now use whenever we do any sequencing. But his research, as he's going to tell us more about today, has been focusing on understanding modifications and different conformations of DNA, such as the G-quadruplexes. And we are really very pleased to have him come and give us his talk today. He's been awarded many prestigious prizes. I won't name them all, but I'll name a few of the sort of key ones. He was elected a Fellow of the Royal Society in 2012. He was knighted by the Queen in 2017. He received the Royal Medal of the Royal Society in 2018, the Millennium Technology Prize in 2020, the Breakthrough Prize in 2022, and the Gardner Prize in 2024. So he's really a very eminent scientist, obviously. It's our great pleasure to have Sir Shankar Balasubramanian here today to give us the Crick lecture. So I think you can come up, Shankar, and start your presentation. Thank you.
Well, it's a great honor and pleasure to be here to give the Crick lecture. Yeah, on a topic that I actually wonder what Francis Crick would think about what I'm about to tell you. Thank you, Madeline and Laurie, for organizing everything today. It's been a lot of fun. What I'm going to talk to you about today actually has some important LMB origins as well, which I'll share with you. And it concerns a structural motif that we call the G-quadruplex. And the history of this field dates back to a paper in 1910 from a German chemist called Ivar Bang, in which he reported an observation that guanylic acid monomers at high concentration formed a gel. So it was implicit that there was some kind of supramolecular chemical interaction that allowed it to form a superstructure. That was 1910. Then nothing happened until 1962, when Gellert at the NIH and David Davis solved by X-ray fiber diffraction what the structural motif was that was responsible for this gel formation. And they showed that monomers, again, monomer derivatives of guanylic acid, formed this four-fold symmetric tetrad, which we call a G-tetrad. And this uses not just the hydrogen bonding that Crick and Watson are famous for, but also the Hookstein edge of the G base at 90 degrees, simultaneously forming 4 hydrogen bonds to adjacent guanines. The other thing this structure showed was that cations could interact in the center. Now, divalents can also bind, but monovalents bind in the order of preference potassium then sodium, and then lithium is the least favorable. And they interact with each of these oxygens, O6 of guanine, uses one lone pair of electrons to form a hydrogen bond, and the other lone pair is available and points into this channel. So this cavity here it has a lot of resemblance to ion channel proteins, actually, dimensionally and electronically. And with sodium, actually, the sodium ion can move through this channel, and synthetic biologists and supramolecular chemists have created ion channels in artificial membranes based on this motif. Now, potassium is the most highly concentrated monovalent in living systems, and potassium is very helpful to stabilize this array. Now, that's a 2D structure based on monomers. If you take a single strand of DNA or RNA, you can form three-dimensional stacked arrays of tetrads. So this is a tetramolecular structure. Or you can form these fold-back structures where a single strand loops back on itself. And this can be done in different geometries. This is an example of an anti-parallel geometry where the strands go up, down, up, down. And this is a parallel geometry where all the strands go in the same direction. And this can also tolerate bulges, rather like is often seen in RNA structures. Now, shown here, this is an example of structural data. This is an X-ray crystal structure solved by Stephen Needle over 20 years ago. And this is for the human telomeric repeat TTA-GGG, 4 repeats in potassium. And you can see the potassium channel here. And this is a beautiful 4-fold symmetric structure in which the TTA loops, the lateral loops, point out to the side. And here you see the 3 tetrads stacked. Now, I first started working on this in the late 1990s. And earlier that decade and in the late 80s, there were a series of papers that use synthetic oligonucleotides of sequences derived from parts of the genome, showing by non-denaturing gel electrophoresis that they could form some kind of higher order structure. And the first of these was a paper from Walter Gilbert and Dipankar Sen from Harvard, and this was using a G-rich sequence from the immunoglobulin region. Subsequent to that, there were papers from Elizabeth Blackburn, Tom Check on telomeric sequences, and also actually here at the LMB, Aaron Klug published a paper on a G-quadruplex inferred by, again, non-denaturing gel electrophoresis. So there began the suggestion that these structures might actually have some physiological relevance in real systems. Now, our work in this area started somewhat by accident, and we were studying human telomerase, trying to understand aspects of its mechanism. And there was a quadruplex aspect because it was speculated that perhaps telomeres could fold into quadruplexes physiologically. And I did a short sabbatical in Bangalore at a place called the Indian Institute of Science. And during that time, one of my students, a chap called Sachin Patel, had a few drinks in the bar at Christ College with his friend, a chap called Mark Ishlan, who's a PhD student from Aaron Klug's lab. And they cooked up an experiment. Some of you may remember that Aaron Klug's lab did some pioneering work on zinc finger proteins, deciphering, in essence, the code of amino acid recognition And zinc finger proteins, of course, they commonly recognize triplets of base pairs through hydrogen bonding interactions in the major groove of DNA. And in Aaron's lab, they'd assembled a zinc finger phage library. And so our work in this area actually started with these two students. After a discussion in Christ College Bar, they did an experiment and selected an artificial zinc finger protein, which they call GQ1, that could bind to a folded telomeric quadruplex with 30 nanomolar KD. So I came back from sabbatical to find some new things going on. And there are students here, and my student came into my office and he said, I haven't been doing what you thought I was doing. I said, oh. And then he presented some really interesting data on what he had been doing. So there's a message in there for all you students about definitely not doing as you're told, because that's when interesting things can happen. Now, they went on to do some experiments to try and see if we could visualize structures in cells using this probe. Now, those experiments didn't progress, but it was actually their work that got me fascinated in the question as to whether these structures could actually form. Now, sometime later, this is around just after 2000, we and others were doing experiments to try and understand the relationship between sequence, structure formation, and also stability of these structures. And I should say, if you take a single strand of DNA and fold it into a quadruplex, typically these are 20 to 25-mers in potassium, physiological potassium and pH. And then you try to melt them so they unfold. The thermal melting temperatures typically range from 50 degrees to close to the boiling point of water. Some of them are very, very thermally stable, even though they're relatively small. Now, I had a student, some of you may recognize because he became a politician sometime after his PhD. In fact, Julian was in my lab, and he was at the Sanger for a while, worked on ENCODE, and then he became a faculty member in the Cavendish for a while, and then he became the MP for Cambridge, and he now runs the Intellectual Forum at Jesus. Now, Julian was, to be honest, he was struggling a bit with experiments, but he was just incredibly bright, and I was trying to find a more suitable research problem for him during his PhD, and so I gave him something theoretical, and he thrived at this and did some amazing work. So Julian wrote a little program based on our understanding of the relationship between sequence and structure formation, taking all the data that was known. And he called this quad parser, and I think this was before it was just after the first draft of the Human Genome reference had come out. So bioinformatics wasn't really a word then. And so I think what he did was quite ahead of its time. And he searched the human genome. I said, search the genome and let's see how many places in the genome there is potential for these types of structures to form. And he found about 350,000 of these motifs in the human genome. Now, this was at a time when papers were being published based on finding one near an interesting gene. So he published this. Now, what became more interesting was looking at the distribution of these motifs with respect to what we then understood about gene structure. What I'm going to focus on today is a relationship between these sequences, the structures, and upstream elements of protein coding genes that are known to be important for the regulation of transcription. And this was the data that really jumped out in this analysis. So this is a probability density distribution. This line here marks the average position of transcription start sites of protein coding genes, and this is upstream and this is downstream. Red and blue are the sense strand and the antisense strand. And by far the hottest spot in the genome is this place here immediately in front of transcription start sites. So this is, the peak is actually around 50 to 60 bases upstream. And there was no strand bias. Well, I'm not going to talk about much today, but downstream, there is a strand bias. And actually there's a peak in the non-template strand. And this is, we believe this is a manifestation of the 5' UTR of messenger RNA transcripts. And there are quite a few papers now showing that these structures can influence what's happening with the ribosome as it's scanning before the translation starts. So these types of observations led to the hypothesis, could it be possible that these structures form in front of genes in one strand? We're not quite sure about the other strand, and there's another secondary structure motif that may form there, called the I motif. Now, I should say the resistance to the possibility of these structures actually existing in the genome was very high. And one of my very strong motivations was being told by some eminent colleagues and friends in the nucleic acids field that I was wasting my time on this useless problem and I should spend my energies doing something more useful. So spurred on by the inspiration of such comments, we kept going. And I'm going to give you some of the highlights specifically around, today I want to focus on promoters and transcription, tell you where we are. We've done a few things with sequencing and adapting sequencing in our lab, and this was a very helpful method developed by former student Vicky Chambers and computational biologist Giovanni Marsico. Going from prediction, we actually figured out how to do a biophysical experiment on the whole genome to map out where these structures could form. The way this works is now with massively parallel sequencing, you fragment the genome and you sequence small fragments on a chip in parallel. And when you have a random sample fragment shown in blue here, you can prime, and the polymerase that is doing the sequencing adds one building block at a time, and each building block gets called by a fluorescence change. So what we do is we carry this out under conditions where the buffer has lithium and not potassium. And lithium is not particularly good at stabilizing these structures. And so we read and record the sequence, and then we melt off this synthetic strand and wash it out. And we reprime and we repeat the experiment. This time we add potassium instead of lithium. We keep it the same ionic strength. Now in potassium, where quadruplexes can form, you have an obstacle to the polymerase at that point. And so the polymerase starts to stutter and starts to make mistakes. So by comparing the first read of this sequence with the second one, you can get this sort of potassium conditional error generation. So you can do this on a genome scale, and in doing so, you can map the start sites of where these structures form. And I should say, things like the DNA double helix, its stability is not particularly sensitive to whether it's potassium or lithium. It's sensitive to ionic strength, but not the nature of the monovalent cation. So this is a structure-specific effect. We can also add small molecules that stabilize this structure and generate a roadblock by that way. So these types of experiments helped us generate experimental maps that were biophysical of where these structures can form. And we've done this now for about 12 model organisms. In humans, we saw a reinforcement of what we saw by prediction, and that is promoters upstream of protein coding genes were the hottest spots where these structures had potential to form biophysically. But we also noted repetitive regions, and of course telomeres, rich in potential as well. So that's a map from purified DNA. Going back to the original experiment we tried in the late 90s to visualize quadruplexes in cells, I should say Hans Lips and Andreas Pluckthorn published the first demonstration of detecting quadruplexes, and this was at telomeres in the ciliate Stylonychia lamellae. And they showed that the telomeres at a certain stage in the cell cycle could form these structures. And Daniela Rhodes from the LNB and Hans Lips did some nice experiments showing that telomere end-binding proteins could regulate the formation of these structures in ciliates. We were focused on human, and one of my students, Julia Biffy, who now runs her own lab at the CRUK Cambridge Institute, she worked with actually one of Greg Winter's former associates, John McCavity, and using phage display of single-chain antibodies, ended up engineering an antibody called BG4 that recognizes DNA G-quadruplexes quite tightly with low nanomolar KD, and with a specificity of 2 to 3 orders of magnitude over double helical DNA. And Julia carried out an early, this was in 2013, experiment in which she used immunofluorescence with this antibody to visualize quadruplexes in cells. So these are different tissue culture cell lines. The nucleus is stained with DAPI in blue, and the red foci represent areas where, using the microscope we used at this time, we could pick up clusters of G quadruplexes. More recently, we've done this at single molecule level, and of course, single molecule fluorescence, you can detect individual features, and we see many thousands of sites in the volume of a nucleus. Now, the other interesting observation she made in the study was the difference between relatively normal cells, so MCF10A is breast epithelial cells, and this cell line, MDA-MB231, is a breast cancer cell line. And we saw persistently more of these structures in cancer cell lines and states than non-cancer. And also looking at cancer normal paired tissue biopsies and staining with this antibody, we always saw much heavier staining in the cancers. So this is an observation. I'm not going to say too much more about this, but feel free to ask me at the end. There is something interesting going on with the formation of these structures in cancer cells. Okay, so at this stage in the story, we can predict, we can detect them biophysically, we can see lots of red dots in an immunofluorescent experiment. And I said to my group, well, this is all good and well, but we're looking at red dots, and that's not totally convincing. These things could be anything. We don't actually know exactly where they form. So I should say in the IF experiments, using chromosome metaphase spreads, we could see that it wasn't just at the telomere. We were seeing foci in the non-telomeric parts of the genome. Now, after many years of trying, one of my postdocs, Robert Hansel-Hirsch, who now runs his own lab in Cologne, he generated the first chromatin maps of these structures, and he used a chromatin immunoprecipitation method with this antibody that was specific for these structures. So quite simply, if you fix cells, fragment chromatin, the assumption is that we're preserving structures and not creating them where they didn't exist. That's an assumption of the method. We then enrich these fragments with the antibody, release the DNA in sequence, and align the fragment sequences to the reference. And you see a topology with lots of fragments being read in particular regions. And our interpretation is that this is where the antibody is recognizing structures in the chromatin. Now, the observations that Robert made was using other methods for detecting relatively nucleosome-free parts of the genome in chromatin. These structures tended to form where the chromatin was relatively nucleosome-free. Now, shown here are the minus strand plus strand outcomes of the sequencing of the isolated DNA. And you'll see in that experiment, there are many places where quadruplexes could form, but in chromatin, most of them do not form. So it appears that chromatin is actually generally suppressing the formation of these structures. However, the observation that he made genome-wide was where the chromatin was relatively nucleosome-free and the transcription was elevated, we saw these structures where they could form. And they also tended to go hand in hand with active proximal histone marks on the nucleosomes. So we published a hypothesis saying that these structures are chromatin features in active chromatin at actively transcribing regions. I should say, subsequent to this early work, we've corroborated these types of observations with a nanobody that we can express in cells that binds to these structures, and we can map out where they bind. And also, as I'll show you, small molecules that can bind to these structures. And there are other techniques some of you will be familiar with using transposition, called cut and tag. And there's also an M&A's method called cut and run. So we've used 4 or 5 quite different methods that all corroborate the initial findings. So we're reasonably confident that this is unlikely to be purely an artifact of any of the components or methods. I'll briefly introduce a method that we call ChemMap that was created by two postdocs in my lab, Zutao, who's soon going off to form his own group at the Genome Institute of Singapore, and Jochen Spiegel, who's in a Cambridge startup company now. Now, as you will know, there are so many drugs that act by interacting with or covalently reacting with DNA. And many of these drugs continue to be used against a broad range of cancers as chemotherapy. Some of them are still used as antivirals. But it struck us that there was no way of detecting their interactions in cells. And we were trying to detect the interaction of small molecules that bind to these structures in cells. And this is one way that we do it. We now have more than one way. If you take a small molecule and you tag it, you can tag it with a biotin, or you can functionalize it with what we call a clickable tag. And you can either incubate it in cells live and do a click reaction afterwards, or you can fix cells and introduce it into the cell. Then you can use either one or two layers of antibody recognition, and you can bring in TN5, which TN5 is a nuclease that cuts and inserts by ligation synthetic adapters. And so you cut out the regions where the small molecule binds. You can amplify these because you've got synthetic primers, and you can sequence, and you can generate, again, maps in the genome that are representative of the binding sites of these small molecules. Here's an example of a small molecule that one of my former postdocs, Rafael Rodriguez, created many years ago. And this comes from a lab in Paris, BenDC3. And so you can create constructs with linkers and an affinity tag biotin. Now, what do we see using these approaches? So this is this molecule PDS, this is BenDC3. You see peaks at sites. If you don't include the small molecule, you don't see these peaks. This is a peak generated by the quadruplex specific antibody. And from the biophysical sequencing, you can see this is the quadruplex that they are picking up. So this is another method of detecting structures, but we also use it to corroborate mechanistically what's going on when we look at the downstream effects of such molecules. So at this stage, we're starting to feel that these structures look like they do form and can form in cellular chromatin. A number of approaches to probe them, and the correlation tells us that the formation might be linked to transcriptional states of genes via some process, some mechanism yet to be defined. So more recently, we've been trying to understand the mechanics of how formation of these structures might lead to transcriptional effects. And I should say there's a wide literature now, growing literature of proteins naturally occurring that have been shown biophysically to be able to bind quite respectively to G-quadruplexes in a test tube. And typically 1 to 10, 15 nanomolar KDs for all these proteins. And there are three types that I want to highlight. One is what we would classically call transcription factors. And I'll say more about this in a moment. Chromatin remodelers that either structurally remodel chromatin or actually epigenetically functionalize histones. And these are the histone modifiers. So these types of proteins have all been found. So Jochen, some time back, he took a look at these ENCODE cell lines, which of course have considerable data logged in the database, to see where quadruplexes form and how this may correlate with the binding sites of these types of proteins. And he made an interesting observation that relates to, I think, a topic that is still not universally accepted, and so there's some discussion and controversy around this. And that is, if you take a population of cells and you ask the question in this upstream element, what are the proteins that are bound here? And this is done typically at a resolution of 200 base pairs. There are a subset of genes that apparently have very many different transcription factors that occupy that site. It can be as many as 150 or 200 that seemingly occupy the same site. So how can that be? Well, there are two heterogeneities. One is across the cell population, all the cells may not be doing the same thing at the same place, and the other is temporal heterogeneity. As we constantly discuss in our group, biology is all about kinetic control, not thermodynamic control. And some of you will know of the single molecule tracking experiments that have shown that transcription factors that bind to sites typically have a half-life of the order of seconds, 10, 15 seconds. So they are dynamically binding on and off in natural biology. So one possibility is transcription factors don't necessarily co-occupy one site at the same time, but they are exchanging, and there's a dynamic flux of them. Now that raises the question mark. Transcription factors, we're told, and the elegant work of Aaron Klug more than 2 decades ago on zinc fingers is that they recognize primary sequence through the major groove. Yet, how do you get so many different factors apparently binding to the same site? So he found a positive correlation whereby those sites where apparently very high numbers of proteins could bind happened to be sites where he experimentally found these structures in the chromatin. So Jochen put forward this hypothesis that maybe there's a structure-based recognition, not sequence-based, structure-based, that could provide a way for lots of different proteins to bind. Now, he tested about 30 of the proteins that he found, and he got evidence for about 25 of them that they do actually bind to these structures biophysically quite tightly. So that seemed to lend some support for this hypothesis. Now, we've carried out a number of proteomic type experiments. I'll describe one. This is from Xiaoyun Zhang. And this is a chemical biology experiment where you take a small molecule, bind it to its target, and you can, through a clickable linker, attach a moiety that is photoactivatable. This is a functionality called a diazarine. If you radiate it, it generates a carbene that reacts instantaneously with any functionality that's proximal. So you can introduce this into cells, allow it to find its target, irradiate it, that will generate the cross-linked proteins, and then you can attach an affinity handle and you can isolate the proteins that are proximal to where this molecule binds. So using approaches such as this, we've detected some interesting proteins that seem to localize at these structures. I'll discuss just 2 of them. One is SMARCA4, which is part of the SWI/SNF motor protein complex that is a chromatin remodeling entity. And this complex is responsible for shunting nucleosomes out of the way to clear space for transcription promoting assemblies to form. And SMARCA4 binds with relatively low nanomolar KD to G-quadruplexes. And if you map where SMARCA4 is located in the genome, we find many, not all, but many of them co-localize at sites where we also detect these structures. Now, this is unpublished data, really, of the last couple of months, generated by Regina Herman, a student in my lab. There are a number of papers that have shown biophysically that this classic zinc finger transcription factor, SP1, which is known to bind to this double helical motif called a GC box in promoters, also binds to G-quadruplex folded structures. There are quite a number of groups now who've made the observation, made the measurements. And so, for example, here are two well-studied G-quadruplexes, and the wild type SP1 DNA binding domain will bind with about 8 nanomolar Kd. And this is the GC box double-stranded consensus, similar binding constant. And this consensus, when flanked with a G-rich context, also generates sites where these structures can form. So there's an overlap between G-quadruplex sites and GC box sites that is experimental, which sort of raises the conundrum, does SP1, this is the classic binding mode for 3-zinc finger protein SP1. We know biophysically that it can bind to this G-quadruplex, rather like the artificial zinc finger protein GQ1 that started all this work in my lab. So the question is, could it recognize either or both of these structures, depending on the context of the situation? Now, I'm going to just give you one piece of data. This is a work in progress, but I thought it might be interesting for this audience. Regina did something, she set out to do something that I said would be really difficult. She wanted to re-engineer SP1 so that it would no longer bind to this double-stranded classic GC box, but would retain its binding for the G-quadruplex. And I said, this is going to be really hard. It's a long shot. Do you really want to spend your PhD doing this? And she said, yes. Now, remarkably, she recently generated a mutant in which she's manipulated finger 3. And this mutant binds to quadruplexes a little bit weaker. It's around 3-fold to 4-fold weaker than wild type, but still respectable, low tens of nanomolar Kd. But it no longer recognizes the double-stranded forms of sequences that have the GC box, higher than micromolar. So there's a big discrimination. And she wants to use this to try and understand whether a protein that looks a lot like SP1 but no longer binds to its canonical motif could do something in cells. Now, I'll show you what she's shown so far. She has mapped the binding site by attaching a flag tack to this mutant. And here are some initial observations. These are in hex cells. SP1 star is the mutant. And nearly all the sites where we detect binding of this were sites where we also detect a G-quadruplex. So it looks a little like this. Here's the mutant protein, and this is where the antibody picks up G-quadruplex structures. And you also get wild-type binding here. Sites that don't have a quadruplex, you don't see this mutant binding. But you do occasionally see SP1 binding because it's canonical duplex sequences there. So I think, you know, we're now in the process of doing complementation experiments to see if we can recreate function via a derivative of SP1 that no longer binds to its canonical sequence. But I think we're open to the possibility that SP1 may also be able to bind to a non-classical structural motif as part of its natural function. Okay, the last couple of experiments I'm going to tell you about are our attempts so far to try and address, following on from the mechanism we think involves protein recruitment, binding to these structures. And these are our attempts to address whether these are sort of causative effects. And this is work carried out by a former student, Isabel, and a former postdoc, Angie. And here we took a very well-studied system in the field, and that is the upstream element of MYC. Now, MYC is one of the most important genes, particularly in oncology. It's a master regulator of many other genes. And it has a very, very complex set of mechanisms for regulating it. Now, we focused on a site where a quadruplex forms very close to the P1 promoter. And our hypothesis is that this structure engages specific proteins as part of what is needed to activate P1 transcription. That's the hypothesis. And so we did a number of genome editing experiments. One was to take the wild type sequence and we mutated out some of the Gs to completely abrogate any possibility of this sequence folding into a G-quadruplex. And I should say there are multiple other stretches of Gs outside this. It's actually quite challenging to take out its ability to form the structure. And then we did a sort of a rescue experiment where we took a different primary sequence known to form a G-quadruplex to see if we could recover any changes that occur when you deplete the quadruplex. And I should say both of these quadruplexes have high resolution structures by NMR and X-ray. They're well studied. So here's the first set of measurements. This is the wild type. Here's where we pick up the quadruplex in the chromatin. And this is the mutant that biophysically can't fold into a quadruplex. And you see the signal goes right down here. And this is the rescue where we put in a different sequence that can still fold into a quadruplex. And you see a recovery in the structure sensing by this antibody. Now, by qPCR, this is a more quantitative view of this. This is wild type, this is the quadruplex deficient mutant, and this is the rescue, a different sequence. Now, when we look at P1 transcription, this is normalized to the wild type. This is the mutant. You see transcription is right down to baseline levels when we take out the structure. And when we bring in this other primary sequence from KRAS and swap it in, we recover the transcription. So this suggests that there's a causative link between the structure formation and transcription. Now, if we look at proteins, here are two proteins that we and others have shown combine to G-quadruplexes, CNBP and SP1. And for both of these proteins, as you go from the wild type to the mutant, you see loss of occupancy, and you see recovery of occupancy when we swap in this alternative primary sequence. So this suggests to us that these types of proteins may be binding to the structure when it's there and playing an important role in enabling transcription. SMARCA4 occupancy at this site also changes. This is the wild type, this is the mutant, and this is the rescue swap. It recovers. Now, a number of labs have looked at the relationship between nucleosome occupancy and where these structures form. And they see what we do, and that is there's an anti-correlation. If you look at the genome, wherever G-quadruplexes are experimentally detected, you see this is a peak of where they're detected. You see a depletion in nucleosomes. So they seem to be mutually exclusive. Now, SMARCA4 is part of the machinery that shunts nucleosomes out of the way. So when we remove the quadruplex, we lose SMARCA4, and actually a nucleosome appears at that site that was not present in the wild type, and I've not shown here, also isn't present in the swap. Now, this is a synthetic biology experiment carried out by Yuqi Chen, who's now at the Sanger in Matt Hurl's lab. And she used the flip-in system of hex cells to create an artificial transcriptional unit. So the thinking here is that if we have a feel for mechanistically what's going on, can we recreate this in a completely artificial transcription unit? So we bring into the chromatin enhanced GFP. Now this is a minimal core promoter that's been used extensively in transcription studies. Now we put in a classical SP1 binding site here as well, because we want to separate the SP1 effect, because we're still not sure, does it bind to duplex or quadruplex or both? And what we've done is inserted in front of the SP1 binding site a number of different quadruplex forming sequences. And here I'm going to show you an example of 3 that are unnatural sequences. This one we call T1 because the G's are separated by a single T loop. Now these quadruplexes in vitro are extremely stable. In relatively low potassium, the melting temperature is above 90. At physiological potassium, you cannot melt them. It's higher than the boiling point of water. If we have 2 T's, it's of intermediate stability, and if you have 3 T loops, it's less stable, 41 degrees TM. Now, if you look at the quadruplex formation using the antibody, you see this level with the T1, this is the most stable quadruplex. This level with the T2, which is intermediate. And the less stable one, we can't actually detect quadruplex formation in chromatin. If you look at the fluorescence, it's high for T1, intermediate for T2, and background for T4. And this is a quantitation of this. This is a G4 negative promoter. This has got T1, T2, and T4. So what this shows is that we are creating transcriptional augmentation by inserting artificial quadruplex forming sequences here in the system. And the quantitative change is directly related to the biophysical stability of what we insert. So this also seems to be in line with what we see with the natural system at MIC. So I'm going to close with this, and I actually gave a talk on this topic probably about 17 years ago here, at a time when I think there was sort of deep skepticism in the world. And so being the LMB, it was a very open-minded audience. And at the end of my talk, where we had very little data then, a group of chromatin people from the LMB came down and said, this has to be a chromatin feature. Well, I agree. I think these structures are features in chromatin. We think they have capacity to recruit key proteins. I haven't talked much about this, but we have other experiments showing that patterns of these structures seem to be linked to cell fate decisions. And we do think now that they play an active role in regulating transcription. I've had many co-workers over the years who've contributed to the methods and other things that I haven't spoken of today, and so I sincerely thank all of those. Most of this work actually was funded by CR UK, so I thank them for their patience. So it's an honor to have given this lecture. Thank you very much.